So hi everyone, good morning. Today it's a nice day to start and learn pathology. So I'm Dr. Chitra. I'll be your Medico Pathology faculty. And today we are going to deal with some questions in pathology. Now, before we begin, there are a few things that I have to tell you. The first thing is workbook for today's class. It has been linked in the description and you can take it. And I would highly suggest that you take a paper and pencil and write down whatever I am saying. Because I believe that it is when you write down that you understand better and that you are able to learn better. So first of all, what is pathology? Now, pathology we know is a study of disease, and you and I and everyone have slept through all the classes in our UG time. But pathology is important because it is one area where you can score marks easily. Now, when we talk about your questions in pathology, you can have three types of questions. The first one being you can have the mugging up type of questions. You know, these are your stains, then your bodies, then your mutations, and then your diseases like such and such is seen in autosomal recessive diseases, such and such is seen in autosomal dominant diseases. All of these are purely mugging questions. You need to mug it up. And I would suggest that you keep a separate notebook for those kind of things, so that you can go through it on the morning of or on the day before your exam, so that it stays fresh in your mind. Second type of questions you have the concept type. Now, these are the things that you need to understand about pathology. And these are the things that are going on in our body. And, you know, these are the questions like, what are the steps of inflammation? Or what is the pathogenesis of a simple thing? The thing is, your question will be, if in case of inflammation, for instance, your options will be increased permeability, decreased permeability, those kind of things. So if you know the concept well, you'll be able to handle those questions. So that is the second type of questions. And you also have a third type of questions, that is the integrated type. Now, this is where you can score when you learn pathology. Now, pathology, as you know, deals with all parts of medicine, including your general medicine, your surgery, your ortho, your OBG. So once you get concepts in pathology right, once you are sure there are some things in pathology, the things in pathology, you'll be able to answer those questions because you have some clue from the history. Now that is the third type of questions, the integrated type. Okay, so today's plan is actually not to make you learn or not to teach you the entire stuff of pathology. The plan is to go through a few of the previous year questions, 100 to be exact, and we will be dealing with those concepts or those things which are important for that kind of questions, and how you should approach those questions, and how you should learn the topics related to that type of question. So I hope it is clear. Again, I like to tell you that your workbook has been linked in the description. So please make use of that. So how we'll be dealing with this? First, I hope everyone has taken out their workbook. We'll look at all the questions. We'll look at questions, and if you're watching live, I would suggest that you put down your answer in the comment box. And then I'll be explaining about the topic, and then we'll come back to the question. I hope it is clear. So moving on, first question. Which of the following is the correct sequence of cellular events of acute inflammation? As you can see, you have 4 options. Now, what is inflammation? Now, inflammation is basically what is happening is, there is homeostasis in the body. All these kind of processes are going on and there is balance. So something happens that disrupts this balance. It may be in form of an organism, it may be in form of ischemia, it may be in form of some toxin, or it may be in form of some environmental effect. So how the body responds is known as inflammation. How the body tries to control that noxious stimuli that is happening, that is called inflammation. Now, in vascularized tissue, you can have several steps of inflammation. First being recognition, recruitment, removal, regulation, and repair. Now, in vascularized tissue, first line of defense is of course the WBCs. Now, the body needs to recognize that an insult has taken place, whatever insult may be. Then it needs to recruit cells so that the inflammation is going to be taken care of. And then it needs to remove that noxious stimuli. And then there needs to be regulation. So now the body is in a state of, I would say, in a state of war against the noxious stimuli, and that has to be brought down to the normal state. So that is your regulation. And then you have to have repair. Repair is, you need to bring the body back to its normal state. Okay. So let us consider a blood vessel. Now in this blood vessel, you have blood that is happening here. Usually what happens is you have leukocytes in the center of the blood vessel, and you have plasma along the sides. So first step is these small WBCs, they have to come out of the blood vessel to the injured site. Okay, how? For that, the first thing is the things in the center, they have to be brought down to the periphery of the blood vessel, and then the blood vessel, the wall of the blood vessel has to undergo some sort of changes so that there is an increased permeability and that these cells can come out. So then third thing what has to happen is, after these cells come out, they should be able to identify where the problem is, and then they have to go to the problem and take care of the problem. Okay, so in that, so we can say first step in inflammation, in any sort of inflammation, is changes in the vascular flow. What does that mean? We have our blood vessels which contain our WBC. Now it is only when there is increased permeability in the blood vessels that WBCs can come out. So that is by vasodilatation. And you also have increased permeability. So what I want you to remember is, if you see inflammation, acute inflammation, it is always vasodilatation, not vasoconstriction. It is always vasodilatation, and it is always increased permeability. Now, this increased permeability, how does this increased permeability happen? These endothelial cells that line the blood vessel, they contract, and they contract. Along with contraction, they release some particles. These are known as histamine, bradykinin, leukotriene, and that is the immediate response. So immediate responses, you have contraction of endothelial cells, release of histamine, bradykinin, and leukotrienes, all of which lead to vasodilation. Now, secondly, suppose there is some sort of endothelial injury. There is some necrosis that is happening. That is also an insult, and also that can lead to these leukocytes coming out. So now we have seen how these leukocytes have come out of the blood vessel. Now, what does these leukocytes do next? Again, the blood vessel, you have your leukocytes in the center. Now, these have to migrate to the endothelium. Okay, so these have to roll and come in contact with the endothelium, and then they have to get attached to the endothelium, and after that they migrate through the endothelium, and then they go to the noxious stimuli. So first you have rolling, you have migration. Migration is, it moves towards the periphery, towards the endothelium. Then you have rolling, and then you have adhesion, adhesion of leukocyte to the endothelium, and then you have transmigration. Okay, this is known as transmigration. That is, it moves through the endothelium to the outside. Now, this transmigration is also known as diapedesis. So, I want you to remember this, because sometimes the option, you will not get a word known as transmigration, you will get diapedesis, and you need to understand both of these are the same. So first, then there are some particles that help in these leukocyte movements. So these are, in rolling, you have selectins. For adhesion, you have integrins. For transmigration, you have CD31, PCAM. Okay. So migration, rolling, adhesion, transmigration, or diapedesis. So rolling you should always think of selectins. Please write it down with me. Rolling, selectins. Now selectins are of three types. You have L-selectin, you have E-selectin, and you have P-selectin. Now, L-selectin is seen with leukocytes. E-selectin is seen on the endothelium. And P-selectin is seen on platelets and on the endothelium. Okay. Now, that is it about selectins. Moving on, we have adhesion. Now, adhesion, when you see the word adhesion, remember integrin. Okay, adhesion is equal to integrin. Integrin. And you have some particles known as LFA, MAC, VLA. So all of this is concerned with adhesion. And thirdly, you have transmigration. And the particle responsible is CD31. It is also known as PECAM1, PCAM1. Okay, I hope this is clear. Now we'll move on to this figure is very important, because this figure has been taken from Robbins, and it is usually seen that in all those exams, like in exams like you have NEET and you have inicet, it's usually the figures that are in Robbins that is taken. So I want you to pay extra close attention to this figure. Here what is happening is, you have rolling, you have adhesion, you have migration. Now, if you can see, this order, the answer is B. I will tell you why. It is rolling, activation of integrin, stable adhesion, and migration. You can see these are the steps that has been highlighted. Rolling, adhesion, migration. I will deal with this in a couple of minutes. So you have rolling, you have the leukocyte coming, you have E-selectin, P-selectin. Then you have adhesion with integrin. Then you have transmigration with PCAM or CD31. Okay. Now, about this activation by chemokines. First, when the leukocyte in blood is not activated, it is just simply going on along its way. When the stimulus comes, when there is something, when the body has decided that there is something wrong in the body releases some chemicals, like, you know, your tumor TNF alpha, your chemokines, and all of that is actually activating the leukocyte, which is how it is getting attached to these selectins. Okay, So first, again, look at this picture, very important picture. If possible, draw it in your workbook. You have rolling, you have adhesion, you have transmigration. And after that, what is happening is you have chemotaxis of leucine. After that, now where have we reached? Now we have reached at this place where the leukocyte has come out of the endothelium and it is now around in the surrounding area, and now it has to go and attach to the noxious stimuli. Again, 1 minute. Okay, so moving on, now you have the leukocyte outside of the endothelium. Now here is our noxious stimuli, whatever it may be. Here you have the noxious stimuli. Now, how does the leukocyte know that this is the noxious stimuli? It needs some sort of identification, or it needs some sort of, something like a signal that this is what you have to do, this is where you have to go. So that process is known as chemotaxis, and it is mediated by your TNF, that is tumor necrosis factor, your interleukins and your chemokines. Okay. Then once the leukocyte has gone here, then what happens? Then it takes care of that noxious stimuli and the cell is free. The cell is free or the body is free. Okay. So here another point that I want you to remember is in the initial stage, that is in the first 6 to 24 hours, your infiltrate is neutrophil. Neutrophil is your first infiltrate, and that happens in 6 to 24 hours. Now in 24 to 48 hours, you have your monocyte. Now is that clear? I will explain this once more. Okay, first you have the endothelium. Here you have a noxious stimuli. In the endothelium, in the blood vessel, what is happening is you have all cells, WBC, platelets, they are all in the center and the plasma is towards the periphery. So, firstly, this leukocytes that are in the center rolls down, attaches to the endothelium, migrates through the endothelium and comes out. Now your leukocyte that is out. Then it moves to the noxious stimuli and it takes care of the stimuli. So here you have rolling, then you have adhesion, then you have transmigration or diapedesis, and you have chemotaxis. Now one step extra, and rolling, it is always by selectins. Adhesion is by integrins, and transmigration is by CD31, otherwise known as PCAM1. Now this is another beautiful figure, again taken from Robbins, which I suggest that you pay close attention to. Here you have a microbe, you have the initial cells, that is the macrophages, the dendritic cells, the mast cells. These come and these give some sort of mediators. Mediators which recruit the leukocytes or the secondary cells, and that causes vasodilation, increased vascular permeability, and these cells come out, and then there is elimination of microbes of dead tissue. This is in a nutshell what is happening in inflammation. So moving on to our question, which of the following is the correct sequence of cellular events of acute inflammation? The answer is B. You have rolling, you have adhesion, and you have transmigration. Okay. I hope it is clear. We'll move on to the second question. Now, in the second question, biopsy from an 8-year-old child with leg swelling showed small round blue tumor cells consistent with diagnosis of Ewing sarcoma. What will be the best method to detect translocation T1122 in this malignancy? Okay, so this requires some sort of explanation. So in pathology, what is the role of a pathologist? What we do is we diagnose things. So how do we diagnose things? We get samples. It may be blood, it may be tissue, or it may be fluids of some sort. Like, you have your pleural fluid, your ascitic fluid, and all. And on the basis of this, we diagnose what the condition is. And it may be non-neoplastic, benign, or it may be neoplastic. So, here you have a boy with a leg swelling. Now, in such a situation, normally either after imaging, normally the surgeon goes for either a biopsy or they will take it out completely. Usually what happens is surgeon goes for a biopsy. So I get this biopsy sample. Now once I get this biopsy sample, there are several things that I can do. First thing is, you process it and you get slide. First thing is I can look for the morphology. Morphology is, you have seen all these cells and all these slides where you have, blue cells, eosinophils, you have neutrophils. Like that, I can try to diagnose a condition by morphology. Suppose in morphology, if I'm not understanding what the condition is, I go for something known as immunohistochemistry. Now here what it is it is essentially a type of staining where I am using antibodies. Now let us take this particular case into account. You have a boy with a leg swelling. So I do the morphology, and what I am seeing is, I am seeing some sort of similar cells. Similar cells. I don't know what these cells are. So what do? I send these for immunohistochemistry. Now in immunohistochemistry, I check for so many molecules. Like you have heard, you have your CD5, your CD4, your CD8. All of these I have stains with me. I stain, and if that cell is taking up that stain, it means it is this cell. Okay, so suppose if it is a T cell, I know it will take CD4 and CD25. Okay, so if I put this cell, if I put stain for CD4 and CD25 in the slide, if that cell will take it up. So I can get an idea of what type of cells it is. So I can have a rough diagnosis. Again, after immunohistochemistry, I still don't know what it is. Another thing that I can do is, I can send the cell for flow cytometry. All of these are new and upcoming things where, again, on the basis of proteins present on the cell, I will be able to identify the cell. Okay. Again, flow cytometry, if I am not able to understand what the diagnosis is, but I have a suspicion. After flow cytometry, it will be like, it can be cell A, it can be cell B, it can be cell C. And then what I do is, I go for fish. In fish, what is happening is, I am having probes that target some sort of translocations, some sort of abnormalities in the chromosome. So if I can understand what sort of chromosomal abnormality is there, I can give a correct diagnosis, because this sort of chromosomal abnormality occurs in this condition only. Okay. Now, even fish is not working, I go for something known as next generation sequencing. Now, why I am telling you all this is, in any sort of question, normally the normal pattern is, the question will say like, a boy or a girl or a short history. These kind of cells are seen in these of cells are seen either in the blood smear or in the biopsy section. And what sort of thing will you do next to confirm? So there is this thing that we follow. First you have morphology, that is the biopsy or the peripheral smear. After that you go for immunohistochemistry. Third thing, if immunohistochemistry is not working, you go for flow cytology, flow cytometry. If that is also not working, you go for FISH. If that is not working, you go for next generation sequencing. I hope you have an understanding of what each of these comprises. Now, we will move on. Here we have an 8-year-old child with a leg swelling, round blue tumor cells, consistent with diagnosis of Ewing sarcoma. So here they have already diagnosed Ewing sarcoma. Now, what will be the best method to detect translocation? I hope you remember me telling you, whenever you need to detect something, detect some sort of translocation, it is always FISH. What is FISH? FISH is fluorescent in situ hybridization. So in any question, you see the word, this is the translocation. What to do next? You always choose FISH. Now here I have some photos of the other option. One, you have this is karyotype. I am sure everyone is very much familiar with this. But in karyotype, you cannot understand if a translocation has taken place because it is all the same color. So you need different colors to identify in this conventional karyotyping. So it can't happen. This is what is known as FISH. Okay, you see images like this, and you see things like fluorescent green and fluorescent red, and once we know which are the probes that is used, we will be able to identify what if the translocation is there or not. Okay, and then this thing, this is a flow cytometry picture. I don't want you to read what is happening here. I just want you to know, if you see such a picture, it is flow cytometry. Okay, and it is a method of identifying cells commonly used in peripheral blood or tissue or whatever. And here you have your PCR, which is the next option here. Now, PCR, whenever you see the word PCR, there are things that you need to remember. Steps of PCR. First is denaturation, then you have annealing, and then you have extension. Okay, so once you see, it is, in fact, it has been asked before in, I think in inicet 2020 or 2021, steps of PCR. It is always denaturation, annealing, and extension. Now, moving back to this question, you have word small round blue cell tumors. Now, this word is very commonly used in pathology, and more commonly it is used in entrance examination. Small round blue cell tumor. There are several types, and I want you to write down examples. Okay. This is some extra thing that you need to learn. Small round blue cell tumors. You have hepatoblastoma. You have medulloblastoma. You have neuroblastoma. You have nephroblastoma. otherwise called Wilms tumor. You have Ewing sarcoma. Then you have retinoblastoma, synovial sarcoma. Now, again, this is an extra point. Please write it down in your notebook and look at it before you enter the examination hall, either on the morning or the day before. Small round blue cell tumor. You have hepatoblastoma, medulloblastoma, neuroblastoma, nephroblastoma, Ewing sarcoma, retinoblastoma, and synovial sarcoma. Okay. So that is all about that question. I hope it is clear.